Good morning, students. Welcome back to the topic replications. Under replication, we have in the last class we have studied about the model. Correct? That is, today we will study about the D-loop replication in mitochondrial DNA. So in mitochondria also DNA is present, right? How they are going to copy, how they are going to replicate, how they are going to form itself one more other copy. Let's see one representation of D-loop replication. Okay, let us start the D-loop replication. So this is a D loop structure in mitochondria. I think you remember the structure of mitochondria. The inner foldings you can see. Okay, here you can see the DNA. Okay, the DNA, how they are going to replicate inside the mitochondria. Okay, so first thing, the mitochondrial DNA, how it is. The mitochondrial DNA, let us discuss some points about this. The mitochondrial DNA is circular. Always it will be in either it may be plant or it may be in animal. They are circular double strand. So you can see here they are circular and double stranded. This is one, this is two stranded. Right? Next. The inner strand is called H strand. Okay, inner strand is called H strand. H means what? Heavy. The meaning of H is heavy. It is a little bit thicker than the outer strand. After this, and outer strand is called as L strand. Okay, which is outer light. L stands for light. Very simple. Mitochondrial DNA is circular and double stranded. Inner strand is called H strand. Outer strand is called as L strand. Okay, L strand. The replication is similar. How the replication takes place? The replication takes place is very much similar. Okay, they are very much similar. That is rolling of circle method. Rolling of circle method. Just they will rotate in the circular manner to replicate itself. So this was explained by Kyrie. This method was explained by scientist called Kyrie. Kyrie explained about this circular when in 1963. Hence it is called as Kyrie's model or D loop replication also. Hence it is called Kyrie model also. Kyrie model. Next, the two strands have separate origin. So here is also origin. Here is also origin. The two strands have the origin is very important. Where replication will start from the origin. Okay. So what we have studied, the mitochondria have double stranded circular ring, and outer strand is called L strand. Inner strand is called as H strand. And both the strands have the origin. Okay, both they have separate origin of replications. Next, the mitochondrial replication, they form balloon-like. So you can see here the balloon-like structure they are going to form. Balloon-like structure appears at the where outer or inner? Outer. You can see. In the L strand, they will form a balloon-like structure. First, during replication, they will form balloon-like structure in the L strand. And this is called as what? D loop, which will form balloon-like structure called as D loop replication. Okay, so the D loop appears opposite to the origin of replication of H strand. Where is H strand and where is the opposite side itself. So here is the origin of the H strand. Opposite to that, the D loop formation will take place in the H strand. Understood? So first two layers are there, H and L strand. The D loop formation will form by forming balloon-like structure exactly opposite to the origin. So here is the origin. Opposite to the origin, the loop, the balloon-like structure, which is called as a D loop. D loop takes place opposite to the origin of the replication of the H strand. Next, the inner H strand. So now the inner H strand function as a template. So these inner H strand act as a template to synthesize the what? Another copy. So now the inner layer, inner H strand act as a template for to synthesize new strand. Inner act as a template to synthesize it. Okay. Now the RNA primer is formed first. Here is the RNA primer is formed. RNA primer is need to replicate. Okay. After this, 3' end, 3' end, RNA primer extended into DNA. Okay. 3', the 3' and the RNA is going to form DNA. Okay, they are going to form the extended into DNA by additional DNA nucleotides. So what happened? Just a simple thing. To synthesize the new strand, nucleotide should be added. Okay. So now the 3' RNA primer, when it is added, they are going to extend it and convert into DNA. How it is going to convert into DNA? By adding addition of the nucleotides. Okay, by addition of the nucleotides. After that, next one, the new strands are going to form the new strands forms the L strand for the template H strand. See here. This is outer as it is. Okay, which is replicating. This is inner H strand, which will replicate and form slowly the new strands. Okay, the H strands will be formed. L strand will be formed. Okay, the L strand will be formed from the H strand. Okay, you can see around the primer when it is formed. The primer is formed and slowly it will circulate and they will form the inner template. So the new strand continue the D loop extended. Okay, as the extension of the D loop reaches at the point, 2/3 round circle will rotate as the RNA primer enters. 2/3 they are going to rotate and form the origin of replication. Now here L strand acts as a template. The new strand forms the H template. See here, this is H, right? H will give rise to L. L will give rise to H by replication. Both act as a template. Okay, by forming the DNA template L strand. Next. So the synthesis of the new strand will be in the opposite direction. This is in this way and this is in this way. See here, opposite direction. This is in the opposite direction. And the beginning, the replication is unidirectional, later it becomes bidirectional. Okay, first it is unidirectional in the beginning. Later it will become bi-directions. Okay, one side and slowly they will cover and they will form up to the origin, terminus and all. So L strand replication is very slow from its original place. Okay, about 100 minutes it requires. It requires 100 minutes. Okay, to replicate. Okay, DNA replication. The synthesis of new strand L is finished over here, then the H. Okay. Okay, L finishes earlier. L finishes replication early. Think this is the H strand, right? This takes this will be late and this will be early. And how much time it is taking? 100 minutes. Okay, this is one more type of the model. Next, we will start with the third model. So in D model, bubble-like structure, we have two strand, H strand, and outer one which is called as what? L strand. Okay, H strand acts as a template and forms the L strand. L strand acts as a template and forms itself the H strand in the opposite direction, and it will take 100 minutes to replicate. Okay, so by forming D-shaped balloon shape opposite to the origin of the thing. Now the third one. Okay, after this was the second model. Next one will be third. Third is linear replication model. Linear replication model of DNA. Of DNA. This is semi, okay, discontinuous bidirectional. Okay, now this is a DNA strand. Already we have discussed. This is one is lagging and another one is what leading strand. So one will be continually formed in this direction. It is bidirectional. Actually, it is a bidirectional. Both the directions they will form the new strand. And here it is not continuous. Hence, it is called lagging strand. We have discussed earlier, and they are in the form of pieces, fragments. So they are called Okazaki fragments, which is not continuous. Okay. Now the DNA, which is going for the real. This DNA of eukaryote T4, T7, and lambda phages are linear. Example, we can see in T4, T7, and lambda phase, which shows linear DNA. Okay, linear is nothing but the not a circular. Okay, linear DNA which is having the single strand. Okay, two types we have circular twisted and linear. Two types were there. Particularly, we can see in the lambda phase, then T4, T7. The DNA replication is either discontinued because it is not going to form the continuous strand there. So it is called as discontinued. Whereas bidirectional 2 with 3 dash and 5 dash direction it is going to form. Okay, and the strand, leading strand, lagging strand also we have discussed, right? Now let us represent the replication eyes. How it is, one representation is there. Let us continue this. So eye formation, eye replication, how it is going to take. Fusion of eye replication. So its origin is there. Wherever origin is there, the eye bulging formation takes place by addition of some enzyme, polymerase enzymes. One we will take, it will form eye-like structure. So eye replication. Now all the eyes start joining, fusing. So fusion of eye replication. You can see the fusion of eye replication. This is the second step. The origins of the replications are involved. Many replication eyes and many replication forks are formed. Let us represent one more this side. So you can see this where the template are the representation of the replication. So here the origins of the replications, the origin, wherever the origin is there the replications are involved and forms many forks. Replication forks formation will takes place. In linear replication, new strand is extended from the RNA primer. So that new strand has an RNA primer at the 5 prime. New strand is present at the 5 prime. Okay, new strand is present at the 5 prime. This is an indication where the always in linear new is present at the 5 dash RNA primer at the 5 terminal also. Konege. Terminal andre konege. At the end, this gap is not filled. Hence, the 5 prime end remains short. So, just very simple. This is the linear. Always the new strand at the terminal of the. This is the new strand. This is the new strand. At the terminal of the 5 prime, there will be new gap. And this will not be fulfilled, okay, and they will remain as a gap itself, okay, they will be remaining as a gap, hence 5 prime end remains short in new DNA, okay, so the in new DNA. Now in the linear model we have seen this, right? What is the coding here? The coding you can see. Think this is a strand. We can write E, T, A, G, G, T, T, A, G, T, A, G, and G I am taking. Okay. This is a telomere of 3 dash, 3 prime. A, P, C, P, C, P, then C. Okay, so this is a 5 dash, 5 prime. So this is a where the coding you can see always 5 prime remains as it is, shorter only. Okay. This will not be there. It will remain shorter in 5 prime. In eukaryote, the terminal end of the DNA are in eukaryotes. Okay, this is in the eukaryote. The terminal DNA are called. The terminal DNA is called in eukaryote. The terminal DNA is called telomere. Telomere means what? Terminal DNA. DNA we will call it as telomere. The telomere contains 6 nucleotides. How many? T, T, A, G. Think this is a RNA template. A, T, C, which is a RNA template. Okay. The telomere have how many nucleotides? 6 nucleotides will be present in the telomere of eukaryotes, and in thousands of repeats. Okay. The repeats do not encode any protein. In thousands of repeats it will take, but no, it will not code for nucleotides. It will not code for proteins. It will not code for the proteins. Okay, next. This is carried by the enzyme telomere, is carried by the enzyme telomerase. Telomerase enzyme is present. Okay, so we have written this E, G, A, G, G's, right? So this will go on adding from the 5 prime. Okay, they will go on adding the 5 prime towards the 3 prime pre-existing. This function acts as a reverse transcriptase enzyme, especially linear reverse transcriptase to synthesize DNA from RNA, HIV. And all we can see, this is a reverse transcriptase enzyme, linear. Okay. So what happened? The telomerase enzyme is added. This enzyme built RNA template carrying the sequence AATCCC. Okay. This will enzyme adds the TT as the TTAGG one after the another. Okay, one after the another to the 3 prime and one after the AATCCC adds to the 5 prime. Okay. Thus, telomerase function as a reverse transcriptase enzyme template. Okay. In the next class, this is in the prokaryotes DNA types of models you have studied. Okay, linear replication model. Next class we will start with the that is in the eukaryotes. Okay, we need some time for representation and all. In eukaryotes, we will study in the next class. What are the enzymes we need? What are that all things we will study later. Same, almost same similar type of Okazaki fragments. They are present. DNA replications almost are there, but in detail we are going to study. Okay, what are the requirements we need? What are the enzymes we need? Okay, we will study detail in eukaryotes. We have studied completed in prokaryotes. Okay, in prokaryotes we have completed. Next in eukaryotes we will study later.